Okay, so the first part, as I had mentioned, we are going through the cell cycle. So, last Friday I discussed the cell, but not really how cells replicate and divide. And the cell cycle is a process in which cells continuously go through a process of growth and division. As more and more cells grow in different parts of the body for different purposes, it's really important to understand this for the MCAT and for understanding how the cell works. There are several phases to the cycle, which I listed here in the bullet points. The first is G1, or the first gap phase. And in this phase, the cell utilizes nutrients and grows larger. It copies organelles and creates building blocks to prepare for the next step. So as you can see here in the diagram, we're starting at the right side here, G1, and then we keep going to here would be the S phase. So the next phase, S phase, is where DNA synthesis takes place, and the amount of DNA is doubled in the cell. And for the purposes of this presentation, I won't go in depth about how DNA replication occurs because we will be going through the process of DNA replication more in depth in a future biology lecture. But for now, we just know that the DNA has doubled in the S phase, right? And after the S phase is the G2 phase, or the second gap phase, in which the cell continues to grow similar to G1, but is preparing for the next phase. So both the gap phases also have checkpoints prior to going to the next step. But usually the G2 checkpoint is more important, especially because it regulates, cell duplication and division. And having unregulated duplication and division can lead to cancer. And so saying the duplication and division, we then go through the next phase, which is the M phase, or mitosis phase, in which the cell divides and undergoes cytokinesis. We will go more in depth into, mitosis and also meiosis in the next slides, but for now, it returns the cells to the original size and contents as it was previously in the G1 phase. And, this cycle, the cell cycle, repeats continuously unless the cell goes into the G0 phase, which you can see it kind of pops up on the side over here. But the G0 phase is the resting phase where cells maintain their size and perform its normal functions usually. And also something to note, you see how there's a smaller circle and then a larger ring. So the smaller ring, I went through each of those phases, but the larger ring has an I, which represents interphase, and that's where G1 or G0, S and G2 phases are making up that portion. And then the M is still an M phase for mitosis. So now, speaking of mitosis, we're going into mitosis. So in mitosis, the first step is prophase, when the genome becomes visible after condensing into chromosomes, the nucleus disappears, the spindle and kinetochore fibers appear, and the central pairs begin to move to the opposite sides of the cell. And so after prophase, which I just was talking about, the next step is metaphase, in which the chromosomes kind of line up at the center of the cell, which forms this metaphase plate. And they do so because the kinetochore of each sister chromatid is attached to the microtubule complex spindle fibers that are attached to, the opposite sides of the cell. And then after metaphase, we go into anaphase, which is when the spindle fibers shorten, as you can see, compared to the previous one, but they shorten and the sister chromatids begin to be pulled apart, and you begin to see a cleavage furrow. It's starting to have an indentation. And then finally in telophase, a nuclear membrane forms around the chromosomes, and then the chromosomes decondense and the nucleolus becomes visible. Yep, okay. I also just wanted to mention that as you can see here, there's the 2N. The cell starts off as diploid in mitosis and then also ends as a 2N, a diploid cell in mitosis. And the daughter cells in mitosis are, you know, exactly identical as the starting ones. So next we're going to go into another process of division, which is meiosis. And you can see I have like a mini diagram here as well. But meiosis is a little more complicated. It's slightly different, but I would say several of the phases occur in a similar manner, not exactly identical, but I'll go into, you know, each of the phases here. So prophase 1 is similar to mitosis because there are condensed chromosomes, the nucleolus disappears, spindle and kinetochore fibers appear, and the centriole pairs begin to move to the opposite sides of the cell. However, the difference is that homologous chromosomes, well, the first thing is that there are homologous chromosomes in meiosis, but that they also pair with each other during synapsis, right? And then after following prophase 1, we have metaphase 1, which also looks similar to metaphase of mitosis, except that the sister chromatids, as you can see in this diagram, they're aligned on top of the metaphase plate instead of like the tetrads being directly like on, like they're aligned on top as opposed to being on that line, except also in the other diagram, it was vertically, and in meiosis here, the first one is like horizontally for metaphase 1. And then also, so after this phase would be anaphase 1, when homologous chromosomes separate from each other while the sister chromatids are still remaining together here. And then afterwards is telophase 1, where the cells are divided here and are considered, by this point, they're considered haploid. So the haploid number, oops, sorry. I'm just gonna, okay. Let me just double check. But okay, let me just go through this quickly. So yes, telophase, we're in telophase, and by this point, the haploid number refers to, there is already a reduction, and N, or the haploid number, refers to the total number of chromosomes found in a gamete. So a sperm or egg cell, for example. And then after telophase 1, so after you have this meiosis 1 portion, we go into meiosis 2. And this is the second cell division of meiosis. That this process occurs more like the process of mitosis, except that they end up with a haploid number of chromosomes instead. As you can see in the diagram, it happens pretty similar. The chromosomes are lining up at the equator, similar to metaphase for mitosis, and you know, they're kind of being separated as well, moving to the different sides. The only difference is that you're going to end up here for meiosis with 4 haploid cells. So, right, we just talked about mitosis and meiosis, but you know, what's the difference between the two? What are the main differences between the two? And so here we have mitosis versus meiosis. So I gave kind of an example here in the diagram. The top portion is mitosis, the bottom portion is meiosis. And because I feel like the MCAT likes to test this as well, like making you guys recognize the differences, I just wanted to drive home these main differences. And so one main one is that cell division occurs once in mitosis and twice in meiosis. If you remember, like there's the meiosis 1 and meiosis 2 part. And mitosis produces 2 identical diploid daughter cells, while meiosis produces 4 non-identical haploid daughter cells. You can see here the top and then the bottom. And then also, as many of you have mentioned, the genetic material is identical in mitosis. Well, that is not the case in meiosis, because in meiosis there's processes like crossing over. And finally, I wanted to mention that in meiosis there is also a halved amount of genetic material from because we turn from a diploid into a haploid. And also mitosis occurs in somatic cells, while meiosis occurs in reproductive cells or gametes.